Hi, welcome back to Bio 131. We are in Chapter 28 talking about eukaryote cells, their origins and diversity. And this is the second of the 3 lectures I'm doing on Chapter 28. I did a quick one, a little bit involved one, that started by just describing what a eukaryote cell was. And you may have skipped that if you've had 130 or Bio 102 before. But for today's lecture, you definitely need to know what is the endomembrane system and nucleus, mitochondria, and chloroplast. And we're going to talk about the origins of these structures in a eukaryote cell. And so following through in our sequence, today what we're going over is the second part here in yellow. And that is we're going to go over the origins of the nucleus and the endomembrane system, the origin of the mitochondria, the origin of the chloroplast, and this is what we call primary endosymbiosis. All right? And in today's lecture, this will be steps 1, 2, and 3. I'm going to do another lecture that will be serial endosymbiosis, and that will be step 4, which I'm not talking about in this little lecture. And then we'll talk about some evidence for step 4 and some other things having to do with the diversity of the eukaryotes and the 4 superclades. But that's for next time. Today we're doing what's in yellow. So talk about the origins of the nucleus and the endomembrane system. And so we've discussed before that there's a relationships between the eukaryote, the 3 domains, eukarya, archaea, and bacteria. And this is what your textbook shows is kind of like a ring of life. And that we have this kind of pool of organisms evolving in very early Earth, about 3 billion years ago. And then what we're going to get about 2 billion years ago is that one branch will come off from there. And it's bringing, in terms of its cell structure, things from the archaea, and then it's going to bring over organelles, basically the mitochondria and the chloroplast, from the bacterial clade. And so basically in the eukaryotes, we're a combination. We are our own domain, but we actually have things from the other 2 domains mixed in with us to make our cell. So simplifying the diagram here, the universal common ancestor, we'll come back to that when we talk about macroevolution. That would be the first cell. From there, it started dividing into basically 2 separate kind of lineages in evolution. One that gave rise to the bacteria, one that gave rise to the eukaryotes. And it's on this lineage giving rise to the archaea. And in that archaea lineage, there's a common ancestor where the eukaryotes separated out from the archaea. So at first you have a lineage going on to the bacteria, a lineage going on to the archaea, which remain as prokaryote cell structure. And we usually define the eukaryotes by its cell structure having membrane-bound organelles. And the 3 biggies are the nucleus and the endomembrane system, the mitochondria, and the chloroplast. And these actually happen in that sequence, 1, 2, 3. And I'll be talking about step 1, step 2, step 3. Going back from a previous chapter, I've just highlighted here, looking on the chart, some of the things in purple let us know that the eukaryotes are more closely related to the archaea than they are the bacteria. And the things in yellow are things that are significantly different from the archaea and actually are used to define the eukaryotes, and those being membrane-bound organelles, the nuclear envelope, right, and differences in our having multiple separate chromosomes instead of the single chromosome loop that we find in the other prokaryotes, right? And so both the bacteria and the archaea have prokaryote cell structure. I'm supposed to say prokaryote up there. And the eukaryote is defined by these organelles and the structure on the inside. Okay, and the way it happened is that we think, we don't know for sure, but the hypothesis is that invagination, folding in of the membranes along the same time that there's an increase in complexity and more genes making separate chromosomes formed the basic eukaryote structure. Then from that, we get the nucleus and the endomembrane system. Secondarily, by this time, the cell can do something called phagocytosis, which I showed you in that little first mini lecture. And so the cell can eat. And hopefully in lab, we've seen amoeba feeding yeast in lab, and you can see the amoeba eating. And so from that, it can do phagocytosis, and then it takes in two very important organelles that are complex. We think the mitochondria came first because all eukaryotes have mitochondria. And then in the second event, we have things that now have mitochondria, right, taking in a cyanobacteria that becomes the first chloroplast. Then we'll have another event I'll talk about later about the other chloroplasts. And so I'm going to refer to these as steps. And we're going to be doing steps 1, 2, and 3 in this lecture. I'll do step 4 in the next lecture. And so first we're going to talk about where did the nucleus and the endomembrane system come from. Step 2, we're going to talk about where did the mitochondria come from. Step 3 will be where the chloroplast came from. And so I want to point out this is following a temporal sequence, right? So step 1, everything has a nucleus and endomembrane system. Then it gets a mitochondria, so now everything has a nucleus, endomembrane system, and mitochondria. And then step 3, some of the ones that have endomembrane system, nucleus, and mitochondria also take in a cyanobacteria that becomes a chloroplast, while other lineages don't. And so that's where we have a division now, where some eukaryotes have chloroplasts and some eukaryotes like us don't have a chloroplast. We only went through step 2. Plants go through step 3. Okay, so remembering I described in the mini lecture, the endomembrane system is a series of membranes. The centermost one surrounds the nucleus with its chromosomes in there. I'll draw some chromosomes, the DNA, and we have separate chromosomes inside. And it connects to the endoplasmic reticulum Golgi and the plasmid membrane to the outside. Mitochondria is an important organelle, and what it does is they convert sugar into cellular energy. The most common form is ATP. And I want to point out that mitochondria are present in all eukaryote organisms, including plants and other organisms that have a chloroplast. So plants use the mitochondria to make ATP just like animals do. The difference is plants use the chloroplast to make sugar to feed their mitochondria. So they have an extra step where they can actually make the sugar to feed the mitochondria, but plants and all eukaryote organisms use the mitochondria to make ATP and fuel the cell. ATP is the common cell energy molecule that we will talk about in different aspects during the course. The chloroplast is the site of photosynthesis, and what it does is uses the energy of sunlight to take in carbon dioxide, fix it, and the end product is sugar, which has calories, it has energy. And then plants and other organisms with chloroplasts can use that to run their mitochondria. So if you notice here in the background, it still has a mitochondria. And this is a plant cell in the Archaeplastida, showing the cell walls and stuff. So it's found in the Archaeplastida, which is the plant kingdom, and red algae. That's one clade. And then in the next lecture, I'm going to talk about how these other groups end up with chloroplasts when their ancestors don't have chloroplasts. And that'd be for the next little lecture. That'd be step 4. Okay, so step 1. How do we get the endomembrane system and the nucleus? And so there's a hypothesis. And so we're going to, I want to make a distinction because we talked about the difference between theory and hypothesis. And remember, a hypothesis is an educated guess. Step 1 is just a hypothesis. Step 2 and 3 and 4 is a theory of endosymbiosis. So there's lots of evidence to support step 2, 3, and 4. We don't know a lot about step 1. But there's some evidence that it happened, but so it's kind of a conjecture at this point, right? It seems the most likely scenario to explain it, but we don't have direct evidence proving it as we do with the others. And so what we have is that you have your original bacteria. Let's try that again. And so I'm going to draw my prokaryote cell here. And originally, a prokaryote cell has a cell wall and a cell membrane. And a single loop of DNA. Okay, over time, this develops some features so it still has its single loop of DNA, but it's lost its cell wall. So no longer has a cell wall. It's developing on the inside a cytoskeleton. And it develops the ability to do phagocytosis. So these are some of the steps that are taking place, right? Again, this is hypothesis, so I'm kind of going over it. Not a lot of evidence to support what's going on here in terms of proof, but it's good conjecture, makes sense of what's going on, kind of steps that must happen until we get to the eukaryote cell. Okay, looking at this, you can see that there's just a little invagination here, kind of pinching in a little bit, and we have the DNA. Still is a single loop. And the next stage in the hypothesis is that the cell folds in on itself with several little patches that may get specialized for other functions. And the DNA is on the inside. And as that happened, maybe when it was dividing by binary fission, the DNA failed to completely separate into two cells. So now you have two chromosomes, and then they can start differentiating, because as one builds up mutations, it starts becoming different from the other. And the next stage would be that now it pinches off, and some of these folds become the different structures, leaving the DNA surrounded now by membrane. And so by invagination, that's folding in. So these are invaginations, and let's say it folds in even more kind of around the nucleus. Well, what will be the nucleus? We now can recognize some of the organelles. We'll now have a nucleus. And so we have our nucleus and endomembrane system. And so this is, for me, what I'll call a proto-eukaryote cell. Proto, because it's before. In your textbook, if you're doing the online quiz for this chapter, they refer to this stage as an ancestral prokaryote. But for me, looking at this stage, it's eukaryote. It's already got these membrane-bound organelles on the inside. So a little bit of technicality between how we're defining them. But I'll refer to this as a proto-eukaryote in all of my quizzes. But if you're doing the online quizzes that come with the Campbell book, the textbook treats this a little differently. Okay, so what I just showed you is this invagination and specialization of the cell membrane. Chromosomes were attached on the inner surface of the plasma membrane, now become attached to the inner surface of what will be the nuclear envelope. And it pinches off, leaving the rest of those little specialized membranes with functions as the endomembrane system. Some of the best evidence we have of this is a couple things. One, looking at key enzymes and other phospholipid membrane proteins and other things at the cell level, we actually see one group of the archaea that is most closely related to the eukaryotes, the locarchaeotes. So that's giving some evidence because we even know within which group they're more closely related. And remember, looking at bacteria, and these are ones that are doing nitrogen fixation, right, and aerobic respiration, we can see that inside here we have the cell membrane that folds in on itself, making these invaginations. And then this I can't even draw, there's so many in there. So we can see that we do have the specialization and invagination of cell membranes in other prokaryotes. So that gives some evidence to it. Okay, so that's our idea of where we get that first proto step 1. We now have our proto eukaryote. And we know that must have happened first because the next step involves phagocytosis. And to do phagocytosis, you need this ability of the membranes to be on the inside, moving as vesicles, moving around. So that whole system of the endomembrane system must have been established before this takes place. Plus, all of the eukaryotes have the endomembrane system. All of the eukaryotes have the nucleus. And so that was a first step in there. Second step is, you can notice this is now taken place, so we have one branch separate from the archaea. Here we have no nucleus. Everything up here now has a nucleus in these two lineages. And we're going to notice that some acquire a mitochondria, and they give rise to all of the living extant eukaryotes. And then we're going to have the second step where one branch, which will give rise to the archaeplastida, will get the chloroplast. So how does this happen? How is this a little bit different from what we've seen before? So in the first system, we had folding in of the membranes. In the second system, what we're going to have is we have a cell that's going to take in another cell, a bacteria. So this is the ancestral eukaryote taking in a bacteria that will become the mitochondria. And then after that, it will take in a cyanobacteria. That will become the chloroplast. Okay, so 1. We have the formation of the nucleus in the endomembrane system from an archaea ancestor. That was our step 1. I'm talking now about step 2, the origin of the mitochondria. Okay, and so in step 1, we have this folding in. So the ancestral prokaryote, that prokaryote here, I want to remind you, is a archaea. Ancestor, not a bacteria. Folds in, and notice the DNA here is inside, and you have these vaginations coming in from the outside towards the inside, and then they pinch off, they break off, they become separate, they turn into organelles. So now we have the nucleus, endomembrane system. That's the ER is kind of drawn the wrong area. ER should be up there. Step 2, we have a lot more evidence for. Right? Same with step 3. And in step 2 is the incorporation of a alpha proteobacteria, is the one that, looking at the genes, is most closely related to, and it will become our mitochondria. Okay, reviewing step 1. Archaea common ancestor has an enlargement in membranes. They fold inside, they invaginate. Different parts of the membrane become different functions. That gives you the endomembrane system. You have the nuclear envelope. You have the ER, rough and smooth ER. You have the Golgi. You have transport vesicles. All those different functions, right? So they specialize. DNA is getting larger. It's accumulating more genes. Probably initially by duplication of chromosomes that fail to separate. So now it's actually adding extra copies of the chromosomes, and then mutation over time can start changing the different chromosomes from each other, and they develop more. Also a possibility that it could have incorporated some plasmids that moved from being plasmids into being a chromosome. So that's that process of increasing the DNA. We know that because the eukaryotes have so much more DNA, many multiple separate chromosomes, where the ancestral archaea must have just had one single loop chromosome and separate plasmids. And this is our nucleus and endomembrane system. Okay, this is our step 1. So we have the DNA. Notice there's no membrane around the DNA. It would be attached at one spot during binary fission. Then it pinches in, separates out, and we get our, what I'm calling the proto-eukaryotic cell from an archaea. Now we're going to do on to step 2. So if we look at step 2, it's taking place after step 1, obviously. And so now we have our eukaryote cell, and it's going to be taken in by phagocytosis basically food. It's eating an alpha proteobacteria. So going back to my diagram here. So we have our beginning of our cell. And so we're going to have our eukaryote cell with its nucleus, put our DNA chromosomes in there. And outside, we're going to have a bacteria. This particular bacteria probably initially had a cell wall around it. Oh, there we go. And this now, our proto-eukaryote, would take it in by phagocytosis. And so the result is it would actually flow around its prey. And I should put the DNA chromosomes in here so we know that's the nucleus. A couple things to notice. So this is taking this in by phagocytosis, but I want you to notice the membrane. So notice how when this is being eaten, it's being surrounded by the plasma membrane of the eukaryote cell. And so when it's taken inside the cell, so it's now called a food vacuole or food vesicle, and it's surrounded by eukaryote membrane, and then inside we'd have the cell wall and the membranes of the bacteria. But rather than dying over time, this cell kept dividing inside the other cell. Sorry about that. And so what we ended up with over time is you would have say multiple of these food vacuoles. And somehow it evaded being eaten, but also the interesting thing about this is being aerobic, where the cell before wasn't. This one with its internal membrane would start actually providing food to the cell. So now cells with this infection would actually be doing better than cells without that infection, because those ones couldn't take advantage of the food being broken down as well as the ones with this. So basically what happens is with this cell, with this inside the cell, it can break down food and get more energy out of it. So if we were to compare two cells at this stage, so this one with the bacteria functioning aerobically inside, this one would be able to get 2 ATP for each glucose molecule, where these ones can get 36 ATP or so per glucose molecule. So thinking about evolution, what's the advantage? What do you think is going to be happening here? You have some cells with this infection, all right, of bacteria growing inside them, but they're getting 36 ATP, that's cell energy, for every glucose molecule that they find in the environment, and the ones without it are only getting 2 ATP. So over time, evolution starts selecting for these, right? So they're better, have a higher fitness. And as they reproduce, this is a very kind of funky mechanism, cell reproducing inside of other cells. These cells no longer have to live and protect themselves from the outside, and so they go through a process of simplification. And in part, they lose their... Oops, let's undo that. Here's our nucleus. And the way I was drawing it, if you notice back here, our bacteria has, I was using orange. So black was the cell wall of the bacteria with peptidoglycan. Inside is its specialized membrane that it's using for aerobic respiration. It loses its cell wall, but it leaves its membrane intact. And so it becomes very reduced, and this now is our mitochondria. So here, they're not really showing it exactly right. They're showing you as if there's a free-living mitochondria outside that was taken up. Obviously, that wasn't the case. Mitochondria can't live with outside a cell. So this must have been a free-living alpha protea bacterial type ancestor that was outside, was taken inside, and then it became simplified and turned into the mitochondria. Okay, so this process we call endosymbiosis. So endo, inside, sim, together, biosis, living. And so it comes about by one organism living inside another organism. And it was a bacteria that was taken up by a young eukaryote cell and over time got simplified and became the mitochondria. Okay, and so this was proposed by Lynn Margulis, and you can read more about it in earlier part in the textbook. So even though I'm talking about chapter 28 for the diversity of the eukaryotes, textbooks kind of assume that you've read chapter 25. So steps 1, 2, and 3 are in chapter 25, and so chapter 28 just kind of starts with step 4 a little bit. So you may want to review that. It's only 2 pages in an earlier part of the textbook, but it's going over what I'm going over right now. All right. This was first proposed by Lynn Margulis about 30 years ago now. And when she proposed it, it seemed kind of far-fetched. And the more and more people studied it, the more and more evidence supported it. And so this is now called the theory of endosymbiosis for the origin of the mitochondria and the chloroplast. And so I just want to point out, this is not talking about the origin of the eukaryote cell. Remember I said that was hypothesis? So we don't know as much about or have great evidence to show exactly how that happens step by step, but there's lots of evidence to support where the mitochondria and chloroplast came from. So that's why we call it a theory, the endosymbiotic theory. So this is step 2. So the eukaryote cell now can do phagocytosis. The cell takes in what we now know today was a common ancestor. The extant or living bacteria most closely related to mitochondria are alpha proteobacteria. It persists inside the cell and continues to reproduce like an infection, but it has a definite benefit to the cell in producing more ATP. And so those cells with the endosymbionts are favored in evolution. They have a higher fitness. They outcompete their neighbors, and so they survive, where other ones just have died away if there are any were surviving ones of these early eukaryotes without mitochondria. And over time, the bacteria that was an endosymbiont living like an infection becomes simplified and becomes dependent upon the cell. Even some of the key enzymes for its function have moved over to the nucleus, and it's turned in now rather an independent cell into an organelle within the eukaryote cell, the mitochondria. Okay, before I move on to step 3, I've modified this diagram, and it doesn't have this lower corner right through here. It doesn't have this arrow. And I want to point out that what I'm showing you here is that from here, step 1 and 2 gives rise to things like the fungi and us, the animals, and the sarclade and the excavata, right? So lots of them only do step 1 and 2. But there's one group that gives rise to the Archaeplastida. That may be cut off there a little bit on the bottom. I don't know if you can see that. It says Archaeplastida. It's one of our clades. Give rise to the plants. So Step 2's already happened. They have a mitochondria, but now we're going to have a very similar process in that they're going to take in a cyanobacteria. And we studied a variety of cyanobacteria in lab. Okay, and so we have our beginning. So this is step 1 up here, right? Making the nucleus and endomembrane system. Step 2, over the second one down, that's getting the mitochondria by endosymbiosis. Step 3 in the archaeplastida is the how they got their chloroplast. Okay, so let's go back. So we leave off, and this gives rise to 3 of our supergroups. The sarclade, excavata, uniconta, and they all have the nucleus and the mitochondria. One group, which is the ancestor to the archaeplastida, those are our plants and red algae. Its name gives it away. Archae means old, like archaeology. Plastid, chloroplast, the old chloroplast. And we'll understand that next when I do the next recording on step 4, where we have a variety of chloroplasts. But this is the first one. And so, again, you have a cell that can do phagocytosis. Nucleus, now mitochondria. And outside, we have a cyanobacteria with its cell wall. It engulfs it by phagocytosis, and initially we would have that same process. Let's get our mitochondria in there. So it would still have a cell wall, and inside would have its photosynthetic membrane. But over time, now he has the benefit is this can actually produce the food to feed the mitochondria. And so that's a huge advantage. Obviously, it's not the only thing that works because we have lots of lineages over here. This gives rise to 3 different groups, all of which have mitochondria and nucleus, but not chloroplasts. This one simplifies. Still has nucleus, still has mitochondria. But now it has an outer membrane that's eukaryotic because it got eaten by phagocytosis. That's that membrane. Loses its cell wall, and now its inner membranes become the thylakoid membranes of the chloroplast. And again, repeating, they have mitochondria because the mitochondria came in back up here. So it's very similar to the origin of how we explain the origin of the mitochondria, the chloroplast is. Okay, step 3. Eukaryote cell already has a mitochondria. That was step 2. An archaeplastida ancestor. The archaeplastida, one of the 4 supergroups in the domain eukaryotes, and it includes the red algae and the plants. So we're talking about the origin of the plants. So the origin of the plant already had a mitochondria, right? Takes in a cyanobacteria. Again, it persists inside the cell, something like an infection, but now it's producing sugar that can feed the mitochondria, which are in the cell, and so this becomes what we call an autotroph, a self-feeder. And so it produces extra sugar, right? And so, again, these ones start out-competing, or definitely are selected for in their lineages, the ones that have chloroplasts, and they survive and evolve. And over time, as we saw in the origin of the mitochondria, we also get simplification. Another thing that lets us know that the chloroplast came after the mitochondria, and probably a good amount of time, is that the chloroplasts aren't as simplified as the mitochondria. Mitochondria can't live without the cell, outside the eukaryote cell, but you actually can make a chloroplast suspension. So if you lyse the cells, you break open the plant cells and release the chloroplasts and give them right conditions, they will continue to grow and divide. All right, and so they are a bit less simplified than the mitochondria. Another indicator that has happened more recently than the origin of the mitochondria. Okay, so now I like this diagram because it's showing you all the steps. So we have our step 1 up here, our step 2, and then in the plants we get our step 3. Right? But notice that they're showing you that there's a whole group of lineages that don't have a chloroplast. This is an extra step that gives us rise to the Archaeplastida. So 1 was the formation of the nucleus. 2 is the formation of mitochondria by endosymbiosis with an alpha proteobacteria. 3 happens in the Archaeplastida, and that's the endosymbiosis with the cyanobacteria. Next time I'll talk about step 4, and those chloroplasts are in lineages that aren't the Archaeplastida, but we need to talk about the supergroups. Okay, so mitochondria descended from a prokaryote aerobic heterotroph, which is an alpha proteobacteria, is the one that's living today, the most similar to it. Chloroplasts are most closely related to cyanobacteria. So what evidence do we have to support that? Well, first of all, we have other evidence of endosymbiosis. And so in coral are an animal, and so when they hatch, they have no chloroplast, they just feed like an animal. And what happens is, as they grow, they will take in algae from the water, and basically they have a layer, think of it as being right under your skin, and the algae live there, and then the algae grow and produce extra sugar and actually feed the animal. So coral actually need these endosymbionts in order to stay alive. And when they're irritated, and particularly when the water temperature changes, they will eat all those endosymbionts, and that's called chloral bleaching because the endosymbionts are what are giving it its color. And they actually have a hard time surviving through that period because some can't get enough food just by living like an animal alone. Here's a smaller, that's a multicellular animal over here. That's a cnidarian, that's a coral. Here's a single cell, what we call a protist. And this, you guys see the axopodia moving out here, right? And this is a radiolarian, and it's actually taking in, and in this case, golden algae cells can persist for some time on the inside. So there's another example we see of endosymbiosis. This is at a multicellular level. This is at a single cell level. Okay, couple things. So if we're looking at it, it's going to be very similar to cell biology because these aren't, we're talking about single cells, so we don't have like bones like you'd see in fossils to show the evidence of what's happened. So we have to look at the cell level, and that means molecular level. So what makes up membranes? And I mentioned before when we're talking about prokaryotes that looking at the phospholipid bilayers and the hydrocarbons and things associated with them and the proteins, you can tell prokaryotic from eukaryotic membrane. Right? Basically, you can tell bacteria from not bacteria membrane. And when I was drawing the organelles, and if I go back and you take a look at chloroplasts and mitochondria, they both have an outer and inner organelle membrane. Kind of drew all over it. But notice here they're showing you in yellow an outer membrane and in orange an inner membrane. And it turns out it's the inner membrane that does all the important stuff. Everything that you associate with the functioning, and that's the way I was drawing it in my diagrams with the orange. And the same with the chloroplast. It actually has several layers of membrane, but the innermost one is the one derived from the cyanobacteria that actually does photosynthesis, and that's what we call the thylakoid membrane. And so if we look at those membranes and their structure, it turns out that the inner membrane is prokaryotic in structure, bacterial, and the outer membrane is eukaryotic. And so how could that happen? Let's go back to my diagram. And so if you look the way I was drawing it, in yellow, I have the eukaryote cell membrane. And notice, as it ate the cyanobacteria, right, the cell wall in black disappeared, but the green plasma membrane, the one doing photosynthesis, survives and becomes the thylakoid. So the outer membrane in a chloroplast, you would expect if it came in by phagocytosis, would be eukaryotic, derived from that food vesicle membrane. And the inside, if it survived, would be prokaryotic. Same if we go back to the story with the mitochondria. It first came in, I don't know if you'd see it, make it a little bit bigger. And we're using orange like in the diagram. And so I don't have to tag it, there we go. So it would be surrounded by eukaryote membrane from being taken in by phagocytosis. Initially a cell wall, and then prokaryote orange membrane doing aerobic respiration. Then as it loses the cell wall, right, you end up with prokaryote membrane on the inside and eukaryote membrane on the outside. So that matches what we'd expect from the hypothesis initially, now theory. Other thing I haven't talked about, and if you get more into cell biology, cells, later on I'll talk about eukaryote cells divide by mitosis and meiosis. And we know that bacteria and archaea divide by a process called binary fission. And so when eukaryote cells, an animal cell or an amoeba cell or a plant cell divide, they divide by mitosis, and that's its nucleus. But the organelles inside the mitochondria and the chloroplast divide independently by themselves by binary fission, just like they did when they were separate free-living bacteria. They have their own circular DNA. And so we like to think of the eukaryote cell as the nucleus having all of the DNA, but it only has the nuclear DNA. The mitochondria have their own DNA, and they use it. They use it to express and make proteins inside the mitochondria. The chloroplasts also have their own DNA. And so mitochondria and chloroplasts have their own DNA. It's in a single loop attached to the plasma membrane, just like a good bacterial cell would. They can use it. So not only do they have the DNA, they have all the mechanisms for transcription and translation, which you may learn about in 130. So basically, they have their DNA and they know how to use it, so it works. Their ribosomes, in order to make protein from DNA, you need a ribosome. The ribosomes inside the mitochondria and inside the chloroplast are the smaller versions of the ribosomes that bacteria have, separate from the ribosomes that would be in the cytoplasm of the eukaryote cell, which are larger ones. And so if you look at antibiotic sensitivity and other things, the evidence is kind of overwhelming that supports this, and that's why we call it a theory. And so it's very clear from multiple levels of study that the mitochondria and the chloroplast came about by endosymbiosis with bacterial ancestry. Other thing I like to point out, to me, it just makes it really clear, because we think, what's the most unique thing about plants? Is they can do photosynthesis. If you took a chloroplast out of a plant cell, it can't make a new one. There's nothing in the DNA of the plant nucleus for making the chloroplasts. Plant cells get chloroplasts from the daughter cells that had chloroplasts. And they can replicate more and more chloroplasts in the cells, but they're passed on. And so that shows you that the Archaeplastida ancestry through coming from the early eukaryote didn't have the ability to do photosynthesis. It acquired it through a cyanobacteria, and it still remains separate in its own little package as a chloroplast. And so that's kind of interesting in that the main feature you think of plants is really bacterial and not plant or Archaeplastida eukaryote information. Okay, so cyanobacteria gives rise to the chloroplast and the Archaeplastida. The alpha proteobacteria gives rise to the mitochondria. I should have flipped that because that's a little out of order. Remember, the mitochondria comes first. The cyanobacteria for the chloroplast comes second. And in the next lecture, we're going to go on and talk about our 4 big clades. But what I was talking about, you can see how the Archaeplastida here is its own separate group. And that's where the chloroplast came from. Before that, we have the mitochondria because everybody's got mitochondria. Okay, so from this lecture at the end, you would want to be able to explain the 3 steps in making a eukaryote cell, including a plant cell. So step 1, making the eukaryote basic cell structure of having the nucleus and having the nucleus and the endomembrane system. Step 2 is how it acquired the mitochondria by endosymbiosis of alpha proteobacteria. That gives rise to these other 3 lineages. And then in the Archaeplastida lineage, you have something that's already eukaryote, already has a mitochondria, acquires a cyanobacteria, which turns into the chloroplast. In our next lecture, I'm going to go over the diversity of these different groups and talk a little bit about another step, a step 4, and talk about mitosis and meiosis.